Hello, I'm Hannah Donner with the Collaborative on Health and the Environment. Che enjoys bringing you the latest environmental health science through our partnerships, calls, webinars, science serves, publications, and social media. I would like to welcome everybody to today's Che Partnership webinar, which is titled Environmental Contributors to Autoimmune Disease, Mechanisms, Impacts, and Chemicals of Concern. Our moderator today is Sarah Howard, founder and manager of diabetesandenvironment.org. We will leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentations, our moderator will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who call in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone in the webinar right now is muted with the exception of our moderator and our speakers. This webinar is scheduled to last for 45 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Sarah.
Thanks, Hannah. Hi, everyone. Thanks for joining us. I'm pretty excited about this call because, well, I have a personal interest in this topic. My son and I both have an autoimmune disease, type 1 diabetes, and having one autoimmune disease puts you at risk for more. So anyway, I'm excited for hearing from our two speakers. We have the first speaker is Dr. Sarah Blossom, who's an associate professor of pediatrics and microbiology and immunology at the University of Arkansas for Medical Sciences. Her research is focused on how the solvent trichloroethylene, otherwise known as TCE, affects the immune system, especially autoimmunity. She's recently co-authored a review on how exposure during development to immunotoxic chemicals can play a role in autoimmune disease. And that review is well worth reading, and it's linked on the resource page for this call. The second speaker is going to be Dr. Jennifer Nyland, who's an associate professor of biological sciences and director of the Henson Honors Program in Science and Mathematics at Salisbury University in Maryland. Her research focuses on the impacts of metal exposures like mercury on the immune system. Some of her articles are also linked on the call page. And she's going to be talking about how mercury can modulate the immune response. So at the end of the call, after both speakers finish, we will have time for questions, which you can type in at any time during the call into the Q&A box at the bottom of the screen. And I will read out those questions at the end. So we're going to start with Dr. Blossom, and you may go ahead.
Okay, thank you. Good afternoon. I would like to thank the Collaborative on Health and the Environment for inviting me to participate in this webinar. And today I'm going to talk about an environmentally induced mechanism that may participate in the immune dysregulation observed in autoimmune disease. Okay, thanks. For today's presentation, I will start with a brief overview of autoimmune disease and focus the discussion on the role of epigenetics. I will conclude with an example and some data from my own lab on our findings of epigenetic changes linked to toxicant-induced autoimmune disease. Autoimmune diseases occur when our immune systems become dysregulated and attack the body's own cells and tissues, resulting in clinical pathological signs of disease in a particular organ or in multiple systems. Autoimmune diseases are quite common, and they affect approximately 5 to 9% of the US population. They disproportionately affect women and are among the leading causes of death and disability in women under the age of 65. There are approximately, to date, about 80 to 100 diseases identified as autoimmune, and some of the more common ones include lupus, type 1 diabetes, inflammatory bowel diseases, rheumatoid arthritis, and multiple sclerosis. And while these diseases are typically classified based on the primary organs that they attack, many share common underlying immunological mechanisms leading to immune dysregulation. Studies have shown that an immune cell called the CD4 positive T cell, in many cases, drive these autoimmune responses. And we do not know why or how autoimmune diseases occur, but we do know that both a genetic and epigenetic, in combination with environmental factors, work together to contribute to autoimmune diseases. Environmental factors are a wide-ranging concept. Both hormonal and endocrine factors play a role, the influence of our microbiome, for example, and exogenous factors, including our diet, medications. However, one other important factor is exposure to environmental pollutants. So our goal is to understand what environmental factors or combination of factors triggers disease in susceptible individuals so that we can target pathways for intervention to normalize immune responses. Because simply removal of the environmental factor is not always an option, especially since we know from studies by ourselves and others that environmentally induced immune altering effects can persist or even manifest later in life after removal of the exogenous factor. So my talk will focus on the interaction between environment and epigenetics. So when contemplating how a toxicant promotes or programs the immune system for autoimmunity, one likely scenario involves epigenetic alterations. Epigenetics literally means above genetics. It refers to modifications to our DNA, the addition of methyl groups or acetylation of histones, for example. And while these modifications do not change the sequence of our DNA, they can affect how our cells read genes, and these modifications are responsible for turning genes on or off. So the science of epigenetics is quite complex, and there are many ways that a cell can alter DNA. And one of the most commonly characterized DNA modification is the addition of methyl groups to a part of the DNA molecule. So these methyl groups bind to cytosine residues in the cytosines bases in the DNA, specifically the ones that are followed by guanine. So they're often referred to as CpG sites. So when a methyl group is added to a cytosine by cellular enzymes called DNA methyltransferases, this prevents transcription factors from binding and genes from being expressed. On the flip side, unmethylated cytosines tend to encourage gene expression because there is nothing to inhibit the binding of a transcription factor. So it's important to note that these changes are regulated by our environment. They are heritable and potentially reversible, which might be important from a therapeutic standpoint. So how does our environment alter epigenetics? From the central dogma of biology, your DNA contains the recipe for making proteins. RNA reads the DNA, and proteins build our cells in our bodies, and cells build tissues, and tissues build our body. However, you interact with the environment in multiple kinds of ways, and a few of them are listed here. So our environment can signal cellular processes to tell our cells which proteins need to be suppressed or activated. And I want to point out that epigenetics is a normal process. For example, in terms of the immune system during infection, promoter regions of certain genes in CD4 positive T cells, for example, become unmethylated so that cells can differentiate into effector memory cells to fight infection. However, in certain diseases, these signals may become dysregulated under circumstances where there may be a less than ideal environment and inappropriately promote or suppress gene expression to promote disease. DNA methylation alterations have been described in several human autoimmune diseases. Because the immune system is very complex, both hyper and hypomethylation has been described. In general, hypomethylation, again, is associated with genes linked to, for example, cytokines and signaling molecules that may promote inflammation. Hypermethylation silences genes that may be important in regulating the immune response. And I've included in this table a few examples. So just to provide an illustration, it's been shown in lupus, certain promoter regions of the CD4 positive T cells are more hypomethylated. It's not clear why, but one scenario may involve the introduction of an environmental factor that by, for example, promoting oxidative stress, may reduce methyl donors and inhibit methylation-dependent enzymes, which would cause these regions of the DNA, of the cytosines, to lose their methyl groups, allowing for transcription factor binding, transcription and translation of genes that have been implicated in autoimmune disease. And studies in humans and also animal models in particular have shown that inhibition of methylation using a drug called 5-azacitidine can induce autoreactivity in otherwise healthy cells. So we know that this mechanism is important in lupus. Okay. So environmental toxicants have been implicated in altering DNA methylation. However, only a few have been studied for their ability to promote immune system activation or autoimmune disease. So I will focus the remaining few minutes showing data from one of these environmental chemicals, trichloroethylene, in which we have shown CpG hypermethylation is believed to play an important role. Trichloroethylene is an organic industrial solvent that is used to remove grease from metal parts. People can be exposed environmentally and occupationally through the drinking water, through vapor intrusion, air, soil, and it is a carcinogen, and it has a variety of, or induces a variety of adverse health effects, including adverse reproductive outcomes. But we also know that it is an immunotoxicant. So it is linked with, in occupational exposures, sort of a TCE hypersensitivity syndrome that involves CD4 positive T cells. We use a mouse model to study TCE-induced autoimmunity where many factors can be controlled. Susceptibility, for example, we use an autoimmune prone mouse model. Females, we can control the dose, the mode of exposure through the drinking water, the timing, whether it's a developmental or an adult-only exposure, and also exposure to other co-exposures. And while we assess target organ toxicity, we are primarily interested in the immunopathology mediated by the CD4 positive T cells to promote autoimmune disease and the mechanisms behind this. Our previous studies have shown that trichloroethylene exposed mice, female MRL mice, these are the autoimmune prone mice in the drinking water, yet have an expansion of these effector memory CD4 cells that secrete increased levels of pro-inflammatory cytokines. They also develop autoimmune hepatitis. And as you can see here, this is T cell infiltration into the liver around the portal vein. We also know from our studies that TCE promotes long-lasting and persistent effects, even if we remove TCE from the drinking water. So the CD4 cells were still altered and the mice still developed autoimmune hepatitis. Because epigenetic effects can persist, we wanted to see if TCE altered DNA methylation. So our lab has conducted 2 studies looking at CpG methylation on a genome-wide scale. There are several techniques, and we use a technique called reduced representation bisulfite sequencing. So we focus on chronic exposures, and these are the 2 studies that we've conducted, a 40-week adult exposure and a 37-week exposure beginning at gestation. So that's a developmental exposure. We isolate effector memory cells from these animals at the end of the experiment, and we isolate the DNA, and we prepare it for our RBS assay. And what this method does is that it enriches for CpG-rich regions of the genome, islands, promoters, and regulatory elements. So I can't show you all of the data, but one of the most interesting findings was that when the differentially methylated regions were compared between TCE exposed and control cells in regulatory elements of the genome, the vast majority, or about 87% of these regions, were hypermethylated. And by looking at the publicly available database, the UCSD genome browser, we were able to annotate these regions, and we found that the majority of these were hypermethylated in regions of DNA that were associated with binding a group of specialized factors called polycomb group proteins. These proteins form a complex called polycomb repressive complex, or PCR2, or PRC2, sorry. PRC2 is typically associated with nucleosomes. Histones. Histones are basically proteins that package the DNA into nucleosomes. Because DNA is so dense, it's a way to compartmentalize the DNA in the cell. The PRC2 complex proteins act as a transcriptional repressor, and they're a group of transcription factors shown here that bind to specific lysine residues, lysine 27 on histone 3. And one component of the PRC2 is EZH2. And it is a methyltransferase that trimethylates these lysine residues in order to suppress gene transcription. The PRC2 complex can also coordinate with promoters of associated genes, as hypomethylated regions tend to attract this complex. So our previous studies show that TCE can increase DNA methyltransferase enzymes in activated cells. So this is how we kind of hypothesized that this is happening. And this is data showing that we know that TCE can activate these methyltransferases. And what happens is this would then methylate the DNA associated with these polycomb group binding sites. And it would thereby inhibit PRC2 binding, so genes that are supposed to be suppressed would instead be transcribed. And others have shown that DNA methylation in these regions can inhibit PRC2 binding. Functional studies by others have shown that by knocking out the gene for EZH2 in T cells where it is no longer expressed, leads to a decrease in regulatory T cells, excessive gamma interferon pro-inflammatory cytokine production, proliferation, differentiation, and enhanced cell survival. Now, this complex has mostly been studied in the context of cancer, but studies in our lab are ongoing to see how this pathway may play a role in TC-induced autoimmunity. So in conclusion, while we acknowledge that autoimmune diseases are complex and multifactorial, we have shown that an environmental toxicant can promote autoimmunity through alterations in DNA methylation in CD4 cells, or possibly. Epigenetic changes may represent an important pathway for autoimmune diseases to develop. And I would like to end by acknowledging my lab and collaborators and sources of funding. And thank you very much for your attention.
Thank you, Dr. Blossom. All right, now we are going to hear from Dr. Nyland. All right.
Okay. There we go. So thanks, Sarah. That was a fantastic talk, and a really, I'm glad we did it this direction because it's a really nice segue into my very specific effects of mercury on the immune response. I have been looking for a while now at how mercury affects the immune system, but I'd like to just sort of put this into context for us. So Sarah did a great job talking about TCE and where it is in the environment. Mercury also is a ubiquitous environmental contaminant, and it's in our water, it's in our air, and it's in our soils. But most importantly, how we are exposed to it as humans is because it bioaccumulates. That means it gets stuck into organisms. And so we have bacteria that make it accessible in the food web, and then phytoplankton or small plants will get it into them, and then the small fishes eat those things, and the bigger fishes eat those things, and eventually we eat the bigger fishes, and then we are exposed to mercury. But we can also be exposed through all sorts of different routes, And one of the ones I want to talk about here is that we've, for a long time, recognized that mercury is an anti-infectant. It has this ability to combat infection or remove pathogens. So humans have used mercury compounds to treat diseases like syphilis and to combat various infections, and used it as either a disinfectant or a preservative. And I don't know whether any of our participants recognize this, but I do remember as a child scraping my knees and having my parents paint them with this mercurochrome. It's a tincture that contains mercury, and it was advertised as being antibacterial and part of the first aid kit, right? But it's mercury exposure. There also used to be calomel, a mercury-containing compound that we could use also as a anti-infection agent. So we've for a long time been exposing ourselves to mercury on purpose, but it's also in compact fluorescent light bulbs, in light switches. It's in your metal mercury amalgams, the dental amalgams. But we also are exposed in ways that we don't know about. What I'd like to point out is that regardless of the kind of mercury that you're exposed to, whether it's those dental amalgams or from light switches, or the version that would be in those fishes that we're eating, the methyl mercury, or ethyl mercury that is used in some of the multi-use vaccines that are out there, although it's been phased out, that all of these have an effect on the immune system. So they are all immunotoxic at some level. And we have evidence using in vitro studies where you have cells in a dish that we're looking at, or experimental models like in vivo animal models, as Sarah was describing, or even studies in humans, epidemiological studies, where we're looking at exposures out in the environment and in populations. But the vast amount of literature that's out there demonstrating that mercury is neurotoxic, so it can make you a little crazy if you're exposed at high doses. Actually, we are only now recently saying at high doses, at low doses, you can also have other effects. But there's a lack of concordance across all of the literature that's out there looking at these different effects of mercury. So we know and we recognize that there are interactions between mercury and other risk factors or susceptibility factors. So some of the things that Sarah was talking about, like the sex of the individual. So women are more likely to develop autoimmune disease. Well, does that mean that they are more at risk from a combined mercury exposure and some other infectious agent or risk factor? And those are some of the things that we're looking at. So in this slide, I'm just showing that we have evidence in the literature to say that mercury is neurotoxic at high doses, but also at low exposures. And that's evidenced in individuals who are co-exposed to mercury in some infection. We also have evidence to demonstrate that there's an increased autoimmune response, evidenced in individuals who are co-exposed to mercury and some trigger. For example, as I'll discuss in a moment, Coxsackie virus B3. And then we also have evidence to demonstrate that in addition to activating the immune response, mercury could also inhibit or suppress the primary immune response. And that's evidenced in studies that are in individuals who are co-exposed to mercury and infection, for example, malaria. So I'll be focusing here on the co-exposure of mercury with an environmental trigger or an infection, in this case, a viral infection. And this is the mouse model that I'll be using. This is the BALB-C model. It's a Coxsackie virus-induced model of autoimmune heart disease. So not one of the autoimmune diseases that Sarah mentioned. In this case, this disease is actually male-typic. So it's more frequently found in males, but it does develop in females. And it's actually a human disease. We believe that the way it works is that some individuals are exposed to Coxsackie virus, and they are unlucky and happen to have the right series of circumstances happening where they will ultimately develop this autoimmune heart disease. So they will become infected with the virus, and during the viral phase of the disease, they will develop inflammation in the heart. So immune cells will migrate and move into the heart, making it inflamed. And just like you would with a flu or another viral infection, you'll feel unhappy and unwell during that time. But most people, most lucky people, are able to have a robust immune response and remove that virus, and the immune cells that have infiltrated into the heart are reduced. And so then that infiltration and inflammation in the heart goes down. Then again, some unlucky individuals will much later go on to develop this autoimmune disease. So we can model that same phenotype that we're seeing in humans, where depending on the strain of mouse that we use, the BALB/c mouse is the one that is this black line. That's a strain of mouse that will develop the viral disease as humans do, be able to clear the infection. They won't see any more virus or any more inflammation in the heart, but then over some time, that particular mouse strain will start to develop an autoimmune disease that, again, those immune cells will start infiltrating into the heart. But at the end of that, it becomes big and flabby, and eventually that mouse will go into heart failure. The red line here is the vast majority of the population of humans who would get the viral infection and clear it, and then they would be fine. So we also have mice that are resistant to developing this virus-induced autoimmune heart disease. What I want you to notice is that both of these, whether they are susceptible or resistant, these types of mice or types of humans, both develop that viral phase of the disease, right? So their immune systems are responding appropriately to the infection, and both cases they're able to clear the infection. The real difference is those later stages. So for mice, that's about 35 days after infection. For humans, it's years later. So what we've done is we have treated the mice with mercury before we've given them the virus exposure. And we're actually infecting them with about the equivalent of a can or two of tuna fish a week. So very low doses of mercury for a short period of time. In this case, it was just 2 weeks exposure. And then let the disease course run its course, and we look at 2 different time points. The day 12 is the peak of the viral phase of the disease. And then we can also look at day 35, which is when we should start to see the peak of the autoimmune phase of the disease. And you'll see also there's this bar that is the interim treatment. So in separate mice, in separate experiments, we also infected them first, let them go through the viral phase of the disease. Once they had cleared it and cleared that infection, we were confident that they didn't have inflammation in their hearts anymore. Then we gave a very short dose of mercury exposure prior to the autoimmune phase of the disease. And we found different results depending on when that mercury exposure occurred. So here are some of the results. We found that the mercury pretreatment, so before they received the virus, was sufficient to change the autoimmune, that later phase of the disease, but not the viral phase. So there was no impact on how much virus was there, how much it was replicating in the heart, or how much the immune system was responding to it and infiltrating the heart during that viral phase, that early phase. So the immune systems of both exposure groups, whether it was control or the mercury in advance of the virus, those responses looked exactly the same. Where we saw the differences was once they had cleared the virus, and we saw that the mercury exposure significantly increased the inflammation that was in the heart. And this bottom panel, the autoimmune viral titer, that's just to demonstrate that the mercury treatment is not activating the virus again. Like, it's not how the virus isn't somehow going latent and hiding in the tissues, and then the mercury exposure is somehow reactivating it. So this is truly an autoimmune response. So it's your own immune system attacking the heart rather than the virus doing the work for you. So there's no virus reactivation, but the mercury or exposure before viral infection is sufficient to significantly increase the inflammation in the heart. And in fact, it was also sufficient to significantly increase the number of mice where we saw this dilated cardiomyopathy, this end-stage heart failure. And you'll notice that the ventricle wall is thin and there's a lot of blue and dark staining in here. That's because there's a lot of immune cells infiltrating and a lot of collagen and fibrosis that's been deposited in that tissue. So it's bigger, it's thinned, it's flabby, it doesn't pump as well as it should. And these mice from the mercury exposure will ultimately go on to end stage heart failure. So we were wondering, how is it that this is happening and what else can we observe? And what we noticed was that there was a lot of that blue staining in the tissue of the heart. When we looked closely, we saw that in fact it was an increased fibrosis that was in the heart in the mercury exposed animals. So again, this is during the autoimmune phase, not related to the viral phase of the disease. And so what could be doing that? Well, there are some other immune cells other than the CD4 positive T cells that Sarah was talking about called macrophages. And these are the cells that you want to have online to help you combat a viral infection. They're part of your innate immune response, and they help to eat up and clear damaged tissue, as well as cells that have been infected with a virus. So we would expect to see a lot of CD4 positive T cells infiltrating into the heart during the viral infection to help remove that pathogen. But we would also expect to see a lot of macrophages infiltrating. Certain versions or flavors of macrophages can, they can change their phenotype and shift slightly. And when they do that, they can become more fibrotic. So those are usually the ones that are helping to repair tissue damage, rather than those that are trying to interact with the virus infected cells and to kill those cells. And so what we were thinking about is that perhaps what's happening is that the mercury exposure is shifting the phenotype or the flavor of these macrophages towards one that is not necessarily appropriate. And it's only happening when the cells, when the mice have been exposed to mercury and that viral trigger. So mercury on its own doesn't do this. It's only when you have the combined exposure. So we looked for the number of macrophages that were in the heart, and we found more during the viral phase of the disease. So you want them there, that's great. But with mercury pre-exposure, they were significantly increased in number. The cytokines were elevated in different patterns than we might have expected. And importantly, there was a lot of fibrosis in those tissues that we think was being driven by this increase in macrophage infiltration. So what does this tell us? Well, the timing of the exposure, so some data that I haven't shown you, is that the timing of that environmental exposure is critical. We did not find any of these same findings when we infected with a virus first and then treated with mercury. There had to be that co-exposure early on in the development of the immune response against the virus, so that pre-treatment with mercury, that timing was critical. If we didn't have that, we did not see the same effects on the autoimmune phase. We saw modulation of these innate immune cells, these macrophages, and evidence of shifting a phenotype toward one that is more pro-fibrotic and less able to, or less likely to be the one that is going after the virally infected cells. So potentially more damaging to the tissue. We also showed evidence of changes at the local site of infection. So in the cytokine milieu, in the signaling environment around these cells in the heart, and those changes in inflammation and then disease severity were evident throughout this at the day 35 time point. So all of these studies were funded by an K99 R00 that I had from NIHS. And so I just want to come back again to why there might be inconsistencies that we see in the literature in terms of the effects of mercury on disease and autoimmunity. If you look closely, there are very big differences in exposure levels. There's differences in the species of mercury that individuals or the different studies are using. So we use an inorganic mercury salt, but what we're exposed to from fish is a methyl version. Ultimately, all of these will become inorganic inside the tissues, but the exposure dose and route and type and species of mercury is important. Interestingly, things like co-exposure, sometimes it's difficult to see what's out there and what actually the co-exposure is, but it could be things like infection. As Sarah has mentioned, co-exposures to different environmental factors, xenobiotics, medications, stress, your genetic background, so those endogenous factors, the sex of the individual, among other things. And so we would say that mercury on its own is not sufficient to cause autoimmune disease, but it's definitely sufficient to interact with other factors to induce some immune-related adverse outcomes. So where are we going next? Well, I've envisioned mercury as this. So on the left, you have this giant speed hump that it's difficult for your immune system to get past on the pathway towards autoimmunity. So normally, we have lots of mechanisms in place to protect us from developing these diseases. But mercury or other co-exposures shave that down so it's much more manageable, like the speed hump on the right. And so then the immune system is dysregulated and can more easily pass over that speed hump and get us onto a path that is pathogenic and detrimental and ultimately ends in disease. So we are actually also thinking about epigenetic modifications, and we're starting to investigate the role of mercury in modulating DNA methylation and epigenetics in macrophages and also T cells. So it's a nice tie-in. The two of us are looking at similar things. And that's what I have. Here's Salisbury University, where I do my work. Thanks.
Great. Thank you, Dr. Nyland. That was great. I'm going to try to share my screen. Okay, anyway, if you have a question, please type it in the Q&A box. We did get an earlier question for Dr. Blossom on, someone asked, what is the time frame for these epigenetic changes to occur? I think it was when you first started talking about the animal studies.
Okay, yeah, I can answer that. We've only done 2 studies, and we've only, unfortunately, when you're working with an animal model, you're only looking at a snapshot in time. So we figured that these epigenetic changes would be more likely to occur over a chronic long-term exposure. So we did our first study. It was 40 weeks of exposure, and that is the time frame that it takes for autoimmune disease to manifest with this kind of chronic low-level exposure. And we also saw very similar results when we started the exposure in early gestation and early life. So, and it amounted to about 37 weeks or so. So we honestly don't know the answer to that question because, you know, we just can't look at multiple time points given the cost of these studies. And I think Jennifer can agree that these kinds of exposure studies are very, very expensive. But what we can do, and what I have been working on, is to try to develop some sort of in vitro system where we can use one of trichloroethylene's metabolites in vitro, just to kind of see, maybe with some methylation specific PCR assays, if it is kind of a quick acting type of exposure that can induce an effect. But in humans and in our animal studies, it's really hard to say how long it takes for these effects to manifest. It's unfortunately an unanswerable question. So, but thank you for the question.
Okay, and then someone asked, can you comment on any hypotheses as to why autoimmune diseases are more prevalent in women? Do toxicants affect methylation or macrophages differently in women, or is it more likely due to different exposures to toxicants?
Jennifer, do you want to take this one?
Yeah, so that's a great question. So some of it is probably related to the hormone signaling and just the general sex differences between the immune response. So men and women's immune responses tend to be a little bit different in terms of how we respond to pathogen, and so then also how we respond to environmental exposure. So the general soup that our cells are all swimming in are different a lot because of the hormones that we're exposed to all the time, just naturally endogenously. And the nature of, you know, at some point you reach an age where you are able to carry a parasite within you for a significant period of time. And so our immune system has to handle that pregnancy scenario, right? So I think also, so just a baseline, men and women are different in their immune systems and the type of immune response that naturally is there and the endogenous system. But also men and women, especially, and also in different cultures, are exposed to different combinations of toxicants because of different, I guess, preferences. So skin lightening, for example. There's a lot of mercury containing skin lightening compounds, and culturally there are different groups that would want to lighten their skin, and that's usually women. So they will be applying those mercury containing skin lightening compounds to their skin. So they're going to be exposed at higher levels to those xenobiotics. I don't know, Sarah, do you have anything else to add to that?
No, I think that answered the question. Just the difference in the immune responses between men and women, I think, is one of the reasons. Mm-hmm. Important reasons.
Okay, and we have a question for Dr. Nyland. What is keeping the macrophages activated? Are NK or NKT involved?
Yeah, that's a great question. We haven't studied that yet. So I know that mast cells are increased in numbers in those hearts as well during the autoimmune phase of the disease and earlier on as well. So both during the viral and the autoimmune phase, we have more mast cells, and those mast cells are more likely to be degranulating. We haven't really looked at T cells. Some of the cytokines that we've looked at, though, would suggest that certainly there's a difference in the CD4 positive T cells and their signaling, but we haven't looked yet at natural killer cells or natural killer T cells. We just don't, right now I don't have the capability to do that here, but that's a great question.
And then one more for you. Is there, someone understands there's a viral component to autoimmune conditions in your study. How are we exposed to CVD3?
Yeah, so various autoimmune diseases have different triggers. And so mine is one example of a particular virus that can, in humans, predispose certain susceptible individuals to developing autoimmune myocarditis. Now, that doesn't mean that it's the only way to get that autoimmune disease, but I will say that Coxsackie virus is one of those viruses that we are routinely exposed to almost annually. It's like the flu, right? You're going to get it, you're exposed all the time. So the question is, if you are one of those susceptible individuals who is more likely to be tipped down that path towards autoimmunity just because of your basic genetics. So you're one of those people who has, like I do, a lot of people in my family with autoimmune disease. So am I more at risk because of my genetics, because of my epigenetics? And then am I also exposing myself to xenobiotics that would, in combination with that viral exposure, push me even farther and shave that speed bump down. It's exciting to think about experimentally and scientifically, but it's this fascinating black box that we just don't know enough about yet.
And then that's all the questions people have asked, but I'm going to ask one more for both of you because I've been wondering this for a long time, which is why we don't see a whole lot of research on a lot of environmental chemicals and autoimmunity.
Lack of funding, I don't know. I think that it's so important, and Jennifer, you can speak to. I just didn't mean to barge in, but these diseases are increasing in prevalence, and I think as we become more industrialized and are exposed to more things, I think they're becoming more and more important. And I think that there needs to be more research. And as researchers go, we want that, but it has a lot to do with the money and how money is prioritized at the funding agencies. Mm-hmm. That's my guess. What do you think, Jennifer?
Yeah, I would definitely agree with that. It's so part of it is the challenge, right? It's complicated. They're complex, multifactorial diseases, and so we can study pieces of them, but though the answers that we get are going to be incomplete, and sometimes that's dissatisfying. And we also might get answers that turn out to be not exactly true when you then combine in all the other things once we're able to do that. So as best we can, I think a lot of us, I think, Sarah, you're probably doing this too, you look in vitro and you look in vivo in animal models, and you also look in NHANES or other epidemiological studies to pull in all kinds of different data to help give you the most complete picture. But it's complicated and it's challenging, but it's also, exactly where is the funding? So what's the hot, sexy new thing that the funding agencies are willing to cover, and what is the public really interested in getting funded? So we have a lot of really good foundations out there that are really pushing to help provide funding to researchers for autoimmune disease research. But you know, if it's a question of cancer versus lupus, they're both devastating, right? But decisions have to be made, unfortunately.
Okay, well, I think we're at the end of the time we have, so I would like to thank both speakers, and I'm going to turn it over to Hannah now to close us down.
Thank you so much for the opportunity.
Thank you.
Thank you.
Thank you both, Dr. Nyland and Dr. Blossom, for your talk. Great. Thank you so much, Sarah, too, for moderating. We're approaching the end of today's webinar. A video recording will be available on SHEA's website soon, and tomorrow you'll receive an email containing a link to the video. SHEA's next partnership webinar will take place Wednesday, May 15th, and is titled Glyphosate, Epigenetics and Transgenerational Inheritance of Disease. To learn more and RSVP, please visit our website at healthandenvironment.org. If you are new to CHE or SHEA and would like to stay updated with upcoming events or more, please sign in to receive our newsletter by selecting the Join Us tab at the top of any page on our website. Additionally, if you appreciate these SHEA partnership webinars, bringing you the latest environmental health research for free, we encourage you to support SHEA's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers for taking time to present today, and to Sarah for moderating. Thank you so much for joining us, and have a great day.